Right.
No, when's that?
It's next Tuesday, but it's in 2.5 hours.
Okay.
So I was going to go next
I'm aware on Tuesday.
I can take
I'm sure something I already forgot because I did we covered that. It's very interesting.
But I think he's targeting, I just find that
But it'll be like stuff like kind of basic. Yeah. Still where
very, very good.
Well, he was here from the coast. Okay, so now you can say herpetologist. He may still be doing his old job too. I by name John's lab came out there.
So if you have
just a warehouse.
I hear him and Jerry almost every morning figure about you don't have a distinct
That is full. Yeah, well, it's like kind of different because it's like old versus coming out like shooting timber. It's like well, you do have to worry about like selling whatever timberland you work at. Well, the officer. Yeah, that's very strong. Yeah, they have the timberland office. And then they also have like
For being here this afternoon. Excited to introduce Dr. Jon Davenport, who's coming to us from the biology department at Appalachian State, where he's an assistant professor. Just arrived there in August. So John's pretty broad in his interests, and you'll see some of that this afternoon. He works broadly in the areas of community ecology, herpetology, Arctic ecology, conservation, and he's also a very creative thinker. One of my favorite papers that he published recently takes a look at inter-kingdom competition between a carnivorous plant and bluegill, which I don't know, is that the only example of competition between kingdoms?
What? So literally 1 month before we got ours out, there was one between spiders and sundew. I know. We don't count that. It's an invert, right?
All right, but anyway, excited to have him here. He's going to talk to us about the effects of climate change on wildlife at the Arctic edge.
Sweet. Thanks, Kyle. Yeah, it's been awesome getting to meet with everyone. I'm looking forward to chatting with folks afterwards. So, like Kyle said, I just got to Boone literally a month ago, so it's kind of nice transitioning back to this area. I'm originally from Sevierville, the Gatlinburg area, so it's cool to be back in the region. So, I'm going to highlight a few of the things that we've been doing in my lab at the very end. So we're all over the map. Like I talked to some of the grad students with some of our interests. I'm going to focus on some of my climate change work that we've been doing in the Arctic, just one, because it makes me look at the data and analyze it and put it together in a framework. So I'm going to show some very preliminary data at one point. I'll stress it's preliminary with some of our population monitoring stuff, but I'm going to focus on some of our different stages. So one nice part about working with amphibians is their land and aquatic phases, right? So we're going to focus on some of the work that we've done with the larval stage of wood frogs at our field sites, some of the adult frogs, and then some of the population monitoring, then kind of wrap it up with some of our overwintering stuff that we're doing. I just got back a few weeks ago from where we're trying to wrap up some of that work, some of our turtle work in West Tennessee, and then some of the new work we're starting in the southern Appalachian streams, really close to Boone, actually. So, to kind of start us off, right, depending on who you adhere to for the news, right, climate change is real or not real, well, it's fairly evident it is real. If you look at the data sets that we have, there's a clear trend with an upward increase in temperatures that's anthropogenically induced, right, so I think we can all agree on that, at least to some degree. And when you think about this, those temperatures and those changes are really not evenly distributed, those anomalies, those changes, right? And so one thing that we do know is there's some regions that are disproportionately affected. The easiest one to talk about, and this is to justify why we work where we do, is the Arctic, right? There's this term called Arctic amplification And you see these shifts. So this is data from 1951 to 1980, and this is how much that deviates from that average data, those anomalies and how they shift. This is just grabbed in February 2016 because you can see some of the extreme ends, but you can see up to 4 to 11.5 degrees C difference from that 30, 40-year mean up in the Arctic. So there's a shift, and a clear shift in some of these higher latitudes, right? And so who gets all the press, right? What do you guys think of when you think of Arctic species?
Polar bears.
Polar bears, right? These guys are the thorn in my side, because when they're at one of my field sites, and they're always there this time of year, that means we can't go work that site because you have to leave it alone for roughly about a day. So I'm not particularly fond of them, but there they are. They're charismatic, right? So this is a female and her 1.5-year-old cub at one of our, really close to one of our field sites. They get all the press because what's happening? Sea ice extent is also steadily declining. So if you look at this data gathered from the National Snow and Ice Data Center collaborative group that puts together sea ice extent, the amount of ice out on the North Pole in the Arctic is declining. And what we're seeing is those ice, not only the amount of ice, but how long it's there. So it's it's drying up, or the ice is melting earlier in the year and it's taking later to form. And so this is a problem, especially for a charismatic species like polar bears, because they're adapted to feed on the ice, right? So they come on shore, they basically starve during those months. They're starving for longer periods of time, or they're becoming opportunistic and eating other things, which could be bad because more interactions with humans, right? So they're getting all this press because of this. And what's interesting is, at our field sites, polar bears get a lot of publicity because we're working up here at the western edge of the Hudson Bay, where some of their populations are declining. And so they've been stable for a while, but they have just started declining in the last 5 years. Some groups out of Alberta have shown. But why I'm interested in not so much the polar bear aspect, but these vast array of peatlands here called the Hudson Bay lowlands. And they're called that because they hug along the Hudson Bay, right? So they're really important wildlife habitat because a lot of birds breed there, especially waterfowl. This is a really nice big breeding ground for waterfowl. But really important for caribou herds, and just a really unique area. And when you also think about it, for me, it's kind of one of the furthest south that you can go and actually experience Arctic conditions. The key characteristic being that continuous permafrost. So here's where our sites are here in Churchill, around there. You can see that continuous permafrost is one of the three defining characteristics of the Arctic. The other being the lack of vegetation, or there's certain types of vegetation, and also those temperatures during the summer, the highs during the summer. And our temperatures here are actually colder than they are over here in Fairbanks, Alaska during the summer. So really do have some of that unique Arctic conditions that go pretty far south. And when you look at it, I like playing around in wetlands, drawn to freshwater environments. It's like a bonanza, you know. So you've got all sorts of these ephemeral wetlands that are here. It's the second largest wetland complex in the world, second only to the Siberian lowlands and other vast peatlands. What's really unique is that you have all this habitat, this tundra, sandwiched in between some boreal forests, and you can see the little fingers here going along this creek. And then you have the Hudson Bay over here. So really in this small stretch of land, you have boreal forest jammed in with tundra, along with the coastal habitat, the Hudson Bay. So it's a really unique area to look at some of these transition zones and some of these unique areas there. And what's happening with us is that we're seeing exactly a similar pattern like you see with the sea ice extent. Our ponds are filled basically from snowpack and from ice out. Ice out is becoming earlier and earlier. So this is one of our ponds next to our field station. This is the old field station. There's a new one right behind you. Old rocket rings that the U.S. abandoned in the 50s. This is one of our ponds with some of the snow still in the spring. This is in late May, and some of the ice that's melting out. So these are opening up earlier, but we're also in tandem with that, seeing them dry earlier. So this is one of our sites out close to the coast. This is the Hudson Bay. There's some rock in this wetland. So you're really seeing the shift, and when the hydro period of the ponds are filling and opening up and drying out, compared to some of our longer-term data. And that's where we talk about the wood frogs, right? So I think just as charismatic as a polar bear, right? Hard not to look at that and think of the poster child for conservation, but to each their own. So wood frogs, super common, really abundant, not at all listed are threatened in any capacity across most of their range. You guys they should be here, right? So locally, so they're here. You're getting near the southern edge of their range. We're up here. They're the only amphibian that goes north of the Arctic Circle. They're the first to breed. When I grew up in the Smokies, you would hear them in January chirping off into the distance. They're like the first ones that'll go over ice and show up and lay their eggs in some of these ponds. Probably live up to 3 years, the oldest, this far south, but we think our populations, they probably live a little bit longer. Probably built because they're frozen a big chunk of the time, so they're charismatic because they're known for being frozen. They have that ability to supercool and basically prevent any sort of ice crystals forming in their internal organs, and so that allows them to persist in some of these really cold environments up here. So, really common species. What we're interested in is like, all right, my students joke about my abilities in PowerPoint, so don't laugh, right? This is a hypothetical larval period for wood frogs, so late May up through about early mid-August. And here's when the pond starts to fill and then starts to dry out. Well, if this is a typical hydro period, what we're seeing is this idea of a phenotypic mismatch. So a lot of great work done in Europe and starting to see some of that in North America where it transitions with birds migrating. And when they migrate earlier, and then they breed earlier, they have eggs, and then those hatchlings come out and their peak abundance of caterpillars corresponds with that peak migration. Well, then that's shifting, and then the food's not available at the same time, so we're seeing some of those mismatches between that. Well, what's kind of happening with us is that hydro period now is shifting earlier, but the wood frogs are not shifting with them. People always ask why that is. We think it's probably because of photo period, not so much temperature that's driving that shift in the wood frogs, probably because the photo period is changing, and it's funny because it's really subtle. You go from, instead of 19 hours of daylight, you go to 18. But that one little shift is something that seems to make them want to go almost to the day you can do. May 29th, May 30th is when wood frogs breed. So we're seeing that shift in the ponds, but not so much the frogs. So this kind of let us ask some of these questions, at least from the larval stage. Well, this could be bad if you're an egg and you're deposited. Can you get out of this pond in time if it's drying earlier? Because we know there's a lot of work that's been done with amphibians that show they can adjust their timing to metamorphosis if a pond is drying, but how about this far north? Can they respond? So we said, well, accelerated drying would reduce survival and their size and metamorphosis, which is a big deal for amphibians because it's linked to future fecundity and reproductive success. We also know we've got data loggers down in the logger ponds that show that these ponds, as they're becoming smaller, tend to become warmer, and they're getting a lot more UV radiation. So it's warming, so we wanted to know, what if you warm up some of these ponds, would there be the ability to compensate, even though they're drying, that warmer, since they're ectotherms, could help drive that shift, to come out in time. So maybe there could be some compensation. So the best way to address this, I like doing experiments, so we set up an experiment. And we set these mesocosms up, roughly about 110 gallons, 416 liters. We could go out and collect the wood frog egg masses. That's what they look like. They attach them to sedge. We could go out and collect them. They're literally all over the landscape. So we could collect them, mix them up, count them out to their desired densities per mesocosm. We add in local pond water and then sedge to give that realism of the actual pond. And we also usually give them a little bit of rabbit chow to help boost the nutrients to get that food web initiated. People like to ask, why are we doing this in winter? That's June. That was a very fun June where we had over 1.5 feet of snow dropped. Right as we're starting the experiment, it was lovely having to break through the ice to put your animals in. So, but these are some of the conditions these animals are exposed to. A lot of variation. We're seeing that with our data set. A lot of weird swings in temperature. So we said, okay, we want to look at drying versus warming. So we set up this 3x3 experiment. And so we said, let's alter the hydro period of the pond. So how often they have water. So for 45 days, 52 days, or 64. The 64 is kind of arbitrary because it's just basically until all the animals came out. That was our permanent pond. The 45 we picked because that's the shortest known larval period for wood frogs from Alaska. So we said, okay, let's push an extreme 45 up to the permanent, which is a little over 60. And we crossed that with 3 different levels of temperature, either ambient, so left them alone, added 1 degree C or 2 degrees C in these mesocosms to try to look at that interaction between drying and warming. And the way that we achieved this change in the warming was adding an aquarium heater, cranking it up and dropping it in. A 50-watt heater gets you about a 1 degree C separation in a 100-watt or a 100-gallon tank, and then a 100-watt gives you about a 2 degree C change. So we had that pretty reliable shift above ambient. What do we measure? Survival, that's pretty kind of neat, right? So survival, we looked at growth of the tadpoles at day 15 and day 30 before we thought they would start initiating metamorphosis. And then we also looked at size and metamorphosis, which, like I said, is the snout bent length for these guys. This is Phil measuring one of the little bitty ones there. And this wood frogs were actually one of the first species of amphibians where we linked size to future reproductive success. So if you come out larger, you're more likely to survive year one, you're more likely to survive the reproduction, and you're more likely, if you're a female, to have more eggs. So it's really important coming out as large as you can early on, right? So diving right into the data, if you look at survival, so when we run our models, this is proportion that are metamorphosing, so basically survival here versus a hydro period. This first one, we see that there is a difference in survival, especially those at that really short hydro period, 45 days, are reduced by over half. So less than 5% survive here, versus we're getting 15 to almost 25% survival when we have that longer hydro period. So we have achieved pushing them to that extreme. They can make it out, but not as many. So this is important for what we're thinking about with shorter hydro periods could be bad. If you warm them, they're more likely to come out again the same way, proportion of metamorphosing, more likely to come out here with these confidence intervals you can kind of see if it's warmer. So if it is warmer, you're more likely to come out. If it's a shorter hydro period, you're less likely to come out. So kind of plays into that. Maybe there is some of that compensation, at least as far as for survival and metamorphosis. And then we look at growth rates. So this first one here is day 15. Really no separation at all, not much going on, right? You look at day 30, we start to see a little bit of a difference coming out with it, and basically what's driving those differences are those day 64 ones, they're not growing as fast, which makes sense if you think about amphibians. They're not getting the cue that, hey, your habitat is drying up. You don't need to hurry to get out, right? So they're slowing down, or at least they're slower relative to the other two. If you look at temperature, same thing, day 15 is here. Not much difference. You're starting to see a trend here at day 30. Those at warmer temperatures are growing a little bit faster. So they are responding to that growing a little bit faster as you add in the temperature, as you heat it up. So then if we look at survival, really the only thing that comes out with this data set is that SVL, those that are coming out at those warmer temperatures tend to be smaller than those at those cooler temperatures. So kind of suggesting that there's a trade-off here. If you can make it out at those, if it's warmer, you're more likely to metamorphose, but there could be that trade-off. You're going to come out smaller. Maybe because you're rushing to get out, and you're getting that trade-off with you're coming out smaller. So what does that mean for the long term for these metamorphs? Probably doesn't look great, but at least they're making it out, right? So they're at least having that opportunity. So it's a lot of stuff to throw, right? A lot of different things there. Kind of to summarize it, like shorter hydro periods, not so good for survival, but warming can help you make it out. Growth rates are increasing as you add temperature, but drying, not much going on, slower. The less they're drying, the slower they're growing. With survival, there could be a trade-off for those that survive being smaller, right? So now, how do you relate this back to polar bears, right? What is the link to polar bears? The link to polar bears, I don't like them, right? So this is a 750-pound subadult male that liked visiting our sites. We tried to do these experiments in enclosures my first year, and pretty sure that this dude is the one that liked to rub his back up on them and destroy them. And he'd way down to the wetland and scratch up against them and then I guess fall over them, or maybe he was so in a trance because they felt so nice that he broke a bunch of them for us. So, but this individual had to be put into polar bear jail and then moved further north up to Nunavut because he came around too often. But I think there is a way that you can try to link this. Now, I don't think the World Wildlife Federation is knocking on my door yet, but I think there is a link because you're like, if these guys are charismatic species for the marine environment, the only really dominant herbivore in a lot of these wetland systems, and they make up a disproportionate amount of some of these ecosystems further north, the only one are wood frogs. We're seeing boreal coarse frogs move into some of our areas, but for the most part, the most dominant herbivore, other than a few copepods, are wood frogs. So these ecosystems, how they're changing, and we have some other experimental data I don't have time to talk about. We're having a lot of nutrient input because snow geese populations are increasing exponentially, and that's increasing the amount of nitrogen and phosphorus going into some of our ponds, And so if you lose the herbivore and you're having these excess nutrients going to these ponds, you could see a dramatic change in some of these ecosystems that we wouldn't have predicted just 15 years ago, 10, 15 years ago. So we think that there is the ability to compensate with wood frogs, but maybe it's not enough. And this change is moving faster than they can overcome, right? So that's where we're kind of at. We have other experimental data I could follow up on, I can answer questions about, but that's where I'm going to stop kind of with the implications here. But one of the fun things that we kept getting asked when we were doing these larval experiments, well, what happens with the adults? And the answer was we don't know. So we were approached by Manitoba Water Stewardship, which actually they just shifted to a different government now in the province. Their Manitoba Sustainable Development and their outlook on how to do things is really different. So the new group don't really care what happens to wood frogs. So luckily I got at least a little bit of funding support when they did care about them in the last provincial government. And they're interested in just knowing, hey, what's going on with these guys, especially when you look at this, you know, the distribution of wood frogs follows that tree line, right? And they're called wood frogs for a reason, because they're found in the woods. They're the most terrestrial ranted frog in North America. So we said, okay, let's figure out where they're going, what they're doing, identify some of their movement patterns, identify some of that critical habitats. So what do they need if they're a charismatic species? What are things that we could do maybe to think about at the adult stage? Because larvae, usually the typical survival for a larval amphibian is about 1 to 2% from egg to metamorph, right? So if they're a lost cause, what do we do to help out the adult survival, right? So why should we care about this? Well, the same thing, remember I talked about we have boreal forest, tundra, and coast all sandwiched in together in less than like 20 kilometers, 30 kilometers. Because what we're seeing, this is some data, some pictures from Europe, but the alpine encroachment of trees. So here you can see the trees in 1999. By 2009, there's definitely more spruce trees. We're seeing that same thing along the polar regions, especially in the north. Here, if you're looking down the top of the globe, the red are those that are increasing in the number of shrubs, so not necessarily trees like the spruce, but at least some shrubby, woody vegetation encroachment. A lot more red there. The blue means stable, but the red means they're increasing. So if you look at across all those 24 sites, you see that the red is showing there's an increase in the amount of shrubs in there. What's really cool about our site is the U.S. government abandoned it in 1959. We had a base there to look at how rockets, how they responded to the Northern Lights, and how that could interfere with rocket launching in case you wanted to get into a war with Russia, right? So once the Cold War was over, we abandoned that area, but what's neat is there are lots of pictures around our field site, and it's all tundra. Now if you go around, the base is almost surrounded by forest. So we have, you know, in a roundabout way, a documentation of, yeah, this used to be more open here. Now it's filled in by some of these spruce trees. So, and you can see that here. So these are some sandhill cranes flying over one of our sites, and the way that they do this is you kind of they follow along these little corridors, but then you have these little tree islands that kind of get situated. Some shrubs get move in with them, and then eventually you see this connectivity between them, and then you have the forest, right? So you see this shifting along, so it's this mosaic of things that are kind of coming along, and there's a lot of folks out of Saskatchewan that are looking at these tree line dynamics. And so for us, we're kind of interested in saying, okay, if this is the Hudson Bay, this is the tundra, and we're sandwiched in between that, you're seeing that shrubby encroachment. Well, what happens with wood frogs? Because they like trees, so simple deductions are they're probably going to like to hang out in the trees, right? So this could be good for them. They could expand and really like this. So we want to look at differences between initially males and females, because there's been a lot of data that's shown there are differences in how females will move further from a wetland because males harass them if they stay close, so females move further away, males stay closer. We thought, why would these be any different there? So we want to look at that micro scale selection, but at the larger scale, we didn't think there'd be much of a difference because they'd probably stay near those trees, so they'd like that spruce or at least that shrubby habitat, right? So how do we do this? We had to collect a bunch of animals, so we go out, and this is kind of looking down, so you can see the density of wetlands here. Looking at one of our sites, these are the roads. This is our site that we call Lindy, that's probably more open, more tundry. This is kind of a transition one here that we call Pulsa. And this is kind of the remnants of the disturbance from the military base a long time ago. And then this is what we call Strange, which is a more forested site. So kind of like more forested, in between, more open. So we grabbed individuals from each one of those sites. We've expanded on this since now. I'm just going to show you these first few years of data. But what do? If we want to follow them, we collect them. From each of those sites, we fit them with a little radio transmitter belt. So this is a individual with a little lap belt. So we have a little 4/10 of a gram transmitter that we put on them, and we track them for 30 to 45 days. So our lives on them are about roughly 45 days, more like 40. And then we record habitat, so we record where they stop along a 1 meter square plot. What are they in? We categorize the vegetation that they're around. And then we also do that at random points along the way. So we have a random coordinate that we choose and go the same distance from the last movement. Look at what that random habitat's like, and we can compare that to what they chose. And so we can generate a lot of those to compare with our logistic regression models. And so what do we have? So across 2 years of data that we have, we've got additional data this year that I just didn't have time to upload and get prepared, but 8, we're nice and even with males and females the first site. The second site, we're a little skewed more towards males. The reason why we didn't actually have this many transmitters, we just had some individuals that would only hold on to it for about 10 or 15 days. There would be some abrasion, so we'd remove them. We didn't want to cause any indirect mortality, so we'd remove them. We'd put them on another individual, so it kind of helped boost up our sample size. The problem was we just didn't have as long with some individuals that first year as we did in the second year, so we had 20 total that we followed for longer. And when we look at these analyses, we threw out any individuals that only had a transmitter for less than 5 days. We didn't look at those because we didn't feel that there was enough data from that period of time. So when we look at that, what do we see? Total distance move. It's not very pretty, but you can basically say there's no difference across these 3 sites across those 2 years when we run our models. There's no significant difference between males and females in the total distance that they move, either also the net distance that they move at any given time. There was no significant difference, but there was at least at one of the sites. This is driven by the few individuals that we have at this one, the few females. Those few females did move a little bit further. So, but nothing really significant enough to where you can say males or females moved really further significantly. So we can lump them in together. And when you just look at the individual frog movements, these are the different colors of different individuals. You can kind of see some really go out and venture, come back. These are the minimum convex polygons. Come out. This one really like to go around the edge of the wetland, would come back over the course of the summer here. So go along and hop around aimlessly like frogs do, right? So we accomplished in achieving knowing that, yep, they hop around a lot. So when we look at the microhabitat scale, so at those 1 meter square plots, we feed all of our things in the model. Each one of these stands for one of these different little microhabitats that we looked at. So moss and lichen, sedge, shrubs, and standing water. We had bare ground too, but we threw that out because there was almost never any bare ground. Only a few times that we had that, and those individuals were ones that we had less than 5 days of tracking. So we threw the bare ground microhabitat out. But when we feed this into the model, our best model, performing model, is that one that includes the water and shrubs and the sedge. And specifically, when you look at it, where do they like to be? Not super far from standing water. Very riveting, right? Frogs, they like water. So again, there's sometimes, ecology and wildlife conservation is elucidation of the obvious, right? So frogs like water, check. We check that box. Sedge habitat, they don't like to be around the sedge habitat so much, but they do like the shrub habitat. So that really low-lying emergent shrub stuff, we do like that. From a larger scale, so we use compositional analysis and looked at this across our sites, and we've since fixed this. One of the things that kind of seemed very obvious right from the get-go, you can see is this is the wetland from where we grabbed individuals originally, and we've said, let's go 400 meters outside from that and characterize some of these, the microhabitat. And when you look at that, most of that's tundra, and only 4% of it was forest. Well, this is our site that's more tundra. Well, when you feed this into the model, where do you think the frogs spend more time? In the tundra. Because there's more tundra, right? So they spend a lot of time in the tundra. So we were surprised at this, even though there's only a sliver there. There's still like those little shrubby areas. We have a transitional thing that we've fixed now. We talk about transitional habitat. It still follows through at this exact site. They still spend more time out in the tundra, less in that transition ecotone, which was still kind of surprising. If you look at our other site, there's a little bit more forest, same 400 meter buffer, 86% tundra. They're spending most of their time in the tundra. And then I think the best representative one, we have a newer field data, like I said, that also kind of confirms this as well. 24% forest. Where are they spending more of their time? Out in this tundra area. Actually out in that really more open. So even though they like at the micro scale, that shrub stuff, they're still more out away from the forest than we would have anticipated. So it's kind of interesting that wood frogs are known for really liking the woods, but here they seem to really spend more time in the tundra. Incidental data that we have that can't really analyze in a shape or fashion, but I can tell you is that we know that individuals that are tagged in the forest and have their transmitters on, we had a higher mortality this last year than we've ever had before of those in the forest than those in the tundra. So could be playing a role. Maybe there are more predators in the forest, especially some of the avian predators hang out a little bit more along that edge, and they'll go out in the tundra and forage, but they will hang out still in the forested zone too. So maybe that's what's driving some of these changes. So adult conclusions, one of the reasons that Manitoba Sustainable Development was kind of interested in this was because of Keystone XL. So now Keystone XL is happening. It's coming through North America, right? It's coming through the U.S., going to Texas. But one of the backups originally, not only going through BC, was coming through up to northern Manitoba and going out at the port at Churchill. And so they're interested to see, like, what are wildlife doing? Mainly they cared about caribou because they're a little more charismatic than wood frogs. Just a little bit. But they're also probably a lot tastier. But they cared about how they would change their behavior relative to the pipeline coming through. But they're also interested in some of the other organisms. So that pipeline's not coming because of other things, but it's kind of interesting because that's what drove some of those initial discussions. But we could at least turn in a report to them, hey, male and female movement doesn't really differ, but that critical habitat that they were using may not all just be boreal forest. And so it's kind of intriguing because you're like, well, you would have expected it would be more forest. There could be positives of them actually really liking that forest and its encroachment, but hey, they're using the tundra already pretty frequently. Some of the implications are that they could be still pushing out those boundaries and expanding and doing fine, better than we had anticipated. And I'll show you some population data that kind of goes along with that in a minute. But some of the future management conservation plans, they were really interested, like I said, in this pipeline, which is not happening, but at least we can provide them some of that data saying, hey, these guys actually aren't just forest dwellers, and they seem to be doing fine in the tundra, at least from a movements perspective. And initially our data with mortality seemed like they were doing just as good in tundra as they were in the forest. Actually, maybe even better. So kind of concluding across these two life stages, tadpoles may be in trouble, but the adults seem to be fine. They're expanding. I think this is a nice part about amphibians, but also kind of the head scratchers. Like you can't just pigeonhole them in one stage or the other. The larval stage may not be doing as well from a climate change perspective because of the shift in the ponds hydro period, but the adults seem to be happy enough. But it can have complex effects on some of these animals. And this is where it's kind of fun to throw a wrench in all that you talk about talking about population level stuff. And we've been doing population monitoring the last 6 years up here across an array of ponds, different sets of ponds. So ponds are dotted all across this landscape, and we have 5 sets of clusters of ponds that we've been monitoring. And so we have some over here in the floodplain of the Churchill River. So this is where the belugas like to come in and calve, and they get shelter away from any predators on the Hudson Bay. We have some out along the coast here, different kind of coastal habitat, more rock, more tundra, more open tundra, and transition areas, and then also over here in more forested areas. So it's kind of spread across about 40 kilometers, and it's kind of fun because you can literally see, here's where the road ends, there's town, there's where the road ends over here. So it's a pretty short area to have to cruise around and sample, relatively speaking, right? So it's a pretty nice gradient, though. Forested here, transitions until you get to the coast, tundra, and the coastal habitat. So we've been going around and counting egg masses. So this is what a wood frog egg mass looks like. They're known for having these big floating rafts, big clusters. Here, usually the biggest that we get is about 20. So this is not our biggest. I think I have a picture of that one later on. But you go through and you can count the egg masses. And what's nice is with wood frogs, if you're going to monitor populations, they make it kind of easy on you. Great work that was done in the early 2000s by groups out of Rhode Island. In the late 90s showed that if you put a drift fence around and count all the males and females that come in, the number of egg masses that are deposited for the most part correspond really well with the number of females. So effectively each egg mass equals one female, and you can relate that back to at least the effective breeding population size of females for this. So it's a good way to indirect measure a population. And it's not very strenuous work, so you just walk around and you look for an egg mass and then you put a toothpick in it. Super high tech, go back. One thing that we learned is don't put toothpicks that are sedge colored because it's hard to see them, right? So this is why it's always fun to find Walmart, the one Walmart that has all the colored ones, blue and red, really stick out really nicely. So we go through and we count these, and we do these usually 3 times. So the first eggs go in. They're known as explosive breeders, so a lot of the range here, they actually spread it out over about 2-week span, 10 days. So we go to at least each side at least 3 times, and like to go and make sure that last time that there's no new eggs deposited, so we can say, okay, breeding is done for this period. Here's what our effective numbers are for this year. So when you look at it, this is extremely preliminary data. I just put these together the other day, so still trying to think about what they are, what they mean. But if you look at it, we've calculated lambda over the last 5 years and looked at the mean daily high in winter. So there's some work done in Michigan that showed that this is important for predicting survival of wood frogs up in Michigan sites where they've had long-term data. And it's interesting because if you look at this, lambda is lower, at least for our floodplain sites, when it's colder. And then it kind of increases up, and then it kind of drops back down as it gets warmer. So there's this kind of like this Goldilocks area in between where it's kind of not too cold in the winter, not too extreme, so mean daily high, not too cold, kind of in between. Same way with our tundra sites. Kind of see that nice weird pattern there where it's like, oh, so here's the R squareds for the floodplain, here's the R squareds for the tundra site. So it's kind of weird. There's like population sizes are changing relative to this. And the same true with the temperature in the fall before they go into winter. We have that same data, you see the same pattern. And you're like, okay, well, this is for a subset of the ponds described by the habitat. And if you look at it, you're like, sweet, it's kind of a generally pattern. Like, so maybe not too cold in winter, not too hot. That's where you might see the biggest change in populations. Then you look at our transition ones, and they're doing the opposite. So what we call transitional, that boreal forests, the tundra transition zones, the lambda for those, when you look at it, seems to drop down the most at those in between. So as it's that Goldilocks period, again, it's like, here it's warmer, they're bouncing back up, but these seem to do better when it's the colder. What I think is driving some of this are just the initial ideas, flushing it out, is snowpack and how snowpack changes. In these transition areas, snowpack can really pile up along those trees and help maybe insulate some of these. And so these over here, really colder, more snowpack, or snowpack data is not as complete. It's a lot harder to get people willing to go out in -20 and measure snowpack for you throughout the summer, or through the winter. Go figure. So we don't have as complete of a data set on that, but I think there's like at least some level of insulation here to where you have colder and more snow could help lead to higher survival and maybe potentially changes in fecundity. So we're kind of interested in seeing how this is going to flush out, but this is only out of our first 5 or 6 years out of doing some of these counts and playing around with this longer term trend. So I think we're doing all right on time, so I'm going to kind of briefly just talk about some of our other projects that we're doing and kind of expand out. So I don't only work in the Arctic. I love the Arctic. It's really awesome. I don't get to spend as much time as I want to there, but there's too many things to try to study in so little time, right? Everybody knows that by now. So some of the things that we're doing is we're expanding out some of our work with wood frogs. And this past, just got back a few weeks ago from setting these up. We're following them to their hibernacula. So this is a little frog condo. This is where that individual decided to stop moving for a few weeks. So he got a little cage put around him, or her, actually it's probably her. Got a little cage put around her, and it's at least a lakeside condo, right? So got a little lakeside condo. We basically put those down. So this is kind of a little closer up, and then you can look down. There's the wood frog. So she actually surfaced when we were putting it in, so we could track them right to where they're at, pull off the transmitter, then put the weigh and measure before they go in over winter, and then we're going to measure them afterwards, and measure fecundity and then change in body conditions, see how that relates to where they choose to overwinter. So we have individuals that we marked, put these out at, and then we have random ones that's also the same kind of idea, like where did they not choose, let's see what those conditions are, how those eye buttons get planted in here with them with a hibernacula, and we can go back and see how they do in either tundra or forest sites. So we're going to try to see if there's any differences between those. We had transmitters on individuals for the transition areas, but some of those transmitters died and we didn't find the frogs at the time. So we have at least some comparisons we're going to do. And on top of that, we're also trying to do this idea of looking, we built these terrestrial pins So we're going to see how they survive the metamorphs, so the young of the year, how well they do when they're trying to overwinter, if there are differences in these forest versus tundra sites, how that would work. And we also have, this is my grad student Tom, who's talking to some local high schoolers who are not at all interested, but we got one of them to try to help set the transects, and they actually did get into it, so it's not entirely true. But he makes these little models that we can look at evaporative water loss, and we can see what the landscape is like as far as for evaporative water loss across these different habitats, and compare that to where the frogs are moving and where they're choosing. So this is kind of the new data that we're trying to collect to try to hone in on what adults, why are they choosing where they're at, what are they doing. So this is kind of stuff that we're fixing to get into. And then further south, so I'm originally from Tennessee, it's hard. I pick a lot of my field sites, with the exception of the Arctic, based on good barbecue. So one of the best places to get barbecue is in Memphis. So it also happens to be that's where there's some alligator snapping turtles. So we've been working with alligator snapping turtles that have been reintroduced back in the early 2000s. Tennessee Wildlife thought it was a good idea to reintroduce them. They decided not to tag or keep track of them. They just stopped at random places alongside the road and dumped them in. And so now we come along later and say, hey, let's see if they're still around. Long story short, 2 out of 73 sites we've been able to find them at. So probably wasn't successful, but there is one, this one, where we've got individuals that are subadults, about 4 kilograms, and we've been tracking where they're moving and what they're doing. 28 individuals so far. They seem to really like just this one little slough. They never go into the main rivers over here. The slough is kind of forested, but it's a weird L shape. They hang out around it, they never really leave, just kind of hang out around there. So it seems like they're doing fine. We know that there's reintroduced ones. This is some genetic output from structure. This is our population of reintroduced ones. This is the ones, like I was telling the grad students earlier, this is called Ballard Slough, but this is a site that we found out because Campbell Soup used to trap them from here in the 60s and 70s and use them for turtle soup. So we know that these are Tennessee ones that have been around for a while. These are Louisiana, northern Mississippi and Louisiana individuals from different populations. And if you look, the same alleles that show up in those show up in our Tennessee ones. So it shows that some of those are introduced. You can look at them too, and they're all like little mini 4-kilogram little clones of each other. So they're the same cohort that were thrown in. So we're following up on some of those, and we're doing some work with mercury too to look and see how much mercury is in some of these guys. And it's interesting because the trigger point for not eating fish is 1 part per million for precautionary advisory in Tennessee for consuming fish tissue, muscle tissue. For common snappers, which are considered a game species and are commercially trapped, there's no limit how many you can have in Tennessee. The numbers for those, so there's a lot of variation, but the numbers are over 4 part per million. So if you have turtle soup, like we talked about at lunch, it can be really tasty. It's probably because of the mercury that's making it so tasty, right? So, but alligator snappers are too. And this shouldn't be that hard to think about either, though, because these two are top predators, right? They're apex predators in these streams because we don't have alligators here, so these guys don't get messed with. So they eat whatever they want, and they're accumulating a lot of that mercury. Sliders don't have as much in them. So we're filling out that work and getting that stuff done. And then the thing that I'll kind of just end on here briefly is we're doing some work with streams, salamanders. So especially moving a boone, it's kind of obvious you need to work in streams, right? So we're switching gears and doing some work with the Tennessee Aquarium. So we set up these aquatic mesocosms that are about 1 meter long here. We have a little sand and substrate gravel that we put in with rocks, and we can fill them through with water. We can manipulate temperature and flow. It's really sweet from an experimental standpoint because we've got field data that we've been doing with non-game fish, and then salamanders, and then crayfish, and we're documenting these changes and how the flows and the temperatures are shifting. But we can also play around with this in these artificial streams and look at things, and what we found is these guys are kind of hard to follow, but the take home is this is an endemic salamander that's only found here in the Cumberland Plateau. This individual is really limited in scope where it's found. It's surrounded by another one called Desmognathus fuscus that's very common, really wide-ranging species. When you warm up these streams, fuscus does not do as well, and this little endemic can outcompete the larger-bodied, more abundant species for a common resource, relative to if it's a cooler stream, then the fuscus will outcompete this individual species, this Abditis, over space and also over food. So it's kind of shifting that interaction between these two as you have these warmer temperatures, and that's what's kind of cool. We expected to find the same thing with these other really common seal salamanders and black bellies. And what's interesting is as the temperature gets warmer, what happens is they actually cohabitate more. So these two species, we thought this individual is usually more stream-side species. It would maybe outcompete this bigger, more aquatic one. Well, it's actually kind of weird. They shift and they hang out under the rocks together. They still are, it increases the more likely of aggressive interactions between the two, so we've got some data from that. But it's weird because they're shifting them to, for some reason we don't know the mechanism, why are they cohabitating more likely when it's warmer temperatures, but they do tend to bite each other, but we don't see any major changes in body condition like we would have expected. So they're cohabitating more, but there's no trade-off, at least with body change, body mass. So lots of fun things that we've been doing with that work. And then I think it's a good place to kind of wrap it up. We're always looking for student researchers, so we have a grad program at App State. So a nice plug. One thing is the Tennessee Aquarium has these fellowships. If students are interested in those, they're really great. They're like REUs that they fund. They have those available. We have grant money. I didn't talk about some of the other work we do to hire technicians. So students that are interested in doing some of that work, we work everywhere from the Arctic to all these other places. My one student is a really great artist that put these together. Really awesome. And these are all the folks. I couldn't have done this without the majority of these people along the way, funding folks that have helped out along the way. And then I'll stop there. I'll leave you with a polar bear for questions. Got to end with that.
Questions, John?
Yeah.
I was kind of curious thinking about the tadpoles. Are most of the ponds on the landscape like uniformly sized, or is there like heterogeneous sizes in the ponds that could act as like refugia while others are drying up?
Yeah, so that's the good and the bad is there's a lot of ponds, so there are refugia. The problem is you get to the bigger ponds that don't dry, we call them, shallow lakes. This is always fun, like this ambiguous term. Like basically shallow lakes, but what do the shallow lakes have in them? Fish. So sticklebacks are there. So we've got some other work that we published that shows sticklebacks can be pretty voracious predators. There's two different species of sticklebacks. So there is a refugia, but the trade-off is you've got to live with a fish predator. So that's why I think it's kind of an intriguing landscape because you have this differing levels of threat, you could call it. You have pond drying, no predators, kind of just a traditional for amphibians. Like lay here, pond may dry, but there's no predators or a different set of predators than this one that's got fish that stays hermit. So we haven't seen a shift. We don't have, but 4 out of our 23 sites have fish at them. So we haven't seen a change in the number really that shift towards the ones with fish with our data set, but it certainly could be the case. But recruitment is probably going to take a hit because they're going to get eaten. So.
For your mercury work, did you look at mercury levels as a function of population density or age in the turtles?
So what we kind of did was we opportunistically took whatever, so we took a toenail and we took a blood sample from each individual and then used that to convert over to actual tissue. And we do have size data. Some of the bigger individuals have more mercury. We haven't really looked at it from a density perspective, the number of individuals. But alligator snappers is pretty easy. There's so few. It might not be as big a deal. And the bad part is common snappers, there's 1 million of them out there. So there's so many, but we could calculate that. We haven't done it yet. So we've had 3 undergrads that have been working on that project, and we're slowly phasing some of that work out as I've transitioned to App State. But yeah, that's certainly a good thing we should do.
I've always thought, but I'm not a hydrologist, but I've always thought that wood frogs were like vernal pool obligates, which might explain their favoritism towards like forests. But has anyone done any work like you have with tracking like resource use of wood frogs in a different region?
So yeah, there's been at least 3 studies that have been done in the temperate zone that show, like, yeah, they hang out around forested. They really like, so this is where our system is really different. And I feel like it's almost like apples and oranges in some ways. But I think it's kind of cool, but it's also not great for comparative purposes. Like, they hang out around wetlands a lot, and they hang out around under leaf litter. And so they really like to hang out around leaf litter around a certain distance from a pond. But we don't have any leaf litter, because all we have is spruce and those little bitty shrubs. So Iron frogs don't move as far as ones that are down in the temperate zone. That's probably a function of there's just so much water across the landscape. Like some of the other work that's been done actually around South Carolina and in Missouri and Michigan, they move further, but there's ponds are further away and where they overwinter at. So I think air animals do things a lot differently too. What's kind of annoying is these guys call all day, so daylight's 19 hours, and you get like 4 or 5 hours of kind of dusky, not true night. They stop calling at night. They call during the day all day. So when you first thing you do, you take somebody up who's a wood frog specialist, like, oh yeah, nothing is at all like what you're used to. You see it laying in these big rafts, these big sections, they don't do that there. So it's like really weird. So I kind of feel like originally I was wanting to do all this comparative stuff, and you're like, of course there's going to be differences because it's so different. So I think that's where it's kind of cool but kind of annoying. But yeah, there have been people that have looked at it, especially the overwintering stuff. People have looked at overwintering in Alaska and showed physiologically the amount that they can handle, the number of freeze-thaw cycles is pretty amazing. And that's what we're kind of wanting to see, what hours if they can handle that same thing. So that's where we're trying to compare it to a similar system. But even there, they don't get as cold as we get. So it's the good and the bad of being in the Arctic with the only amphibian kind of out on an island. So, yeah.
All right, well, quick announcement. We're going to go out after seminar here to back streets, so change of venue from the usual, but hopefully you can join us there and listen to Jon M.
Okay, good. I'm glad that you got to play in it. And you had fun. And you really was.